damage, then repair, and clinical significance of, I mean, if any damage happens to the DNA, so what type of disease you will get? Okay, maybe usually what happens when the DNA is damaged? I mean, when the DNA is damaged, usually what happens? It may lead to the mutations, right? Mutation lead to cancer, early aging, apoptosis, cell death may happen. Then you will get a lot of hereditary disease also. So, lot of clinical significance are there when there is a DNA damage, right? So, DNA in cells is continuously exposing to the, I mean, endogenous and exogenous agents also. So, what are the endogenous and exogenous agents? See, for example, if you uh, wander in sunlight, even the sunlight also, even the sunlight also, or if you are exposed to ultraviolet rays directly, okay, or if you are exposed to some chemical agents, then automatically your DNA structure may be altered, right? See, for example, if you talk about the free radicals, I think day after tomorrow you'll be having a class on the free radicals, okay, reactive oxygen species we call ROS. So those will cause the oxidative damage, okay, like a superoxide radicals, hydrogen peroxide, hydroxyl radicals. All these will affect the, the base pair modifications, okay, and also they do the break in the DNA, okay, so they may cause the cancer or neurodegenerative, I mean degenerative disease they will cause. So a lot of things will happen, right? So we have exogenous factors, endogenous factors, even the food substances also. Okay, they may cause the cancer because some may be alkylating agents. Some are, for example, you take the nitrous acid. Okay, when you take the nitrous acid, then that will cause the deamination. Okay, I mean, lot of bacteria, viruses also. They may cause the, DNA rupture, okay, or DNA damage. Okay, so a lot of factors are there. Got it. So usually what we do, we will try to keep the DNA structure as it is. Because what is the DNA here? DNA is nothing but the genetic information, right? So you are passing your genetic information to your offspring. Means you are giving your complete characters to the future generations, right? So if you are passing the damaged DNA, it leads to the mutation, right? It leads to the mutation. Got it. So this DNA will not be protected for future generations, right? If it is not repaired. So that is the reason the DNA should be repaired continuously. If there is any error in the DNA base pair or if there is any damage in the DNA, then you have to repair it immediately. So how to repair the DNA? I mean the damaged DNA. So initially we will be learning about how many types of damages are there and what are the factors for the DNA damage. Okay. Then after learning the damage, then we will be talking about the mechanism of repair. So how to repair means anyway we are not going to repair our body only. It will do the because in our body we have a lot of enzymes and proteins are there, right? So they will do the modification or they will help in the mechanism of DNA repair. Okay, so how many types of repairs are there? So these things also will be discussing in this DNA damage and repair. Okay, and if it is not repaired, then you will get the diseases, right? So what type of disease you will get? Maybe hereditary disease or cancer-like symptoms also you will get. Okay, and okay, so yeah, so this is about the introduction. Okay, now we'll see in detail. Yeah, see here, the diagram is explaining, right? DNA damage, how the DNA is getting damaged here, cellular metabolism, exposure to UV light, using the ionizing radiations like UV rays, X-rays, also gamma rays, they may cause the DNA damage, right? Then chemical exposure, replication errors. What's the replication error? So in the last class we discussed about the replication, the DNA replication. So at the time of replication also, if there is any replication error, so that should be addressed and that should be repaired, right? So that is called a proofreading. There's a word called the proofreading. So what it will do, it will check once whether the replicated DNA is exactly as it is like a parent DNA or not. So it will do the proofreading. While doing the proofreading, if there is any error, then the exonuclease will cut that part, and the endonuclease, I mean, usually it will, or polymerase, it will repair that part, right? So if there is any replication error, then it will also causes the DNA damage. So these are all the different ways to damage the DNA. Okay. Then what happens here? Cell cycle checkpoint activation, transcriptional program activation, then DNA repair, direct reversal. When all these things will happen. So when there is a DNA damage, what happens? Say apoptosis, the cell may die. This is one reason. Or sometimes when the DNA is damaged, then direct reversal will happen using the enzymes and proteins in our body. Direct reversal means the repair mechanism. Then base excision repair also will happen. Means whatever wrong base is connected, or wherever the wrong base is there, so it will excise that, it will cut that base and it will repair that one. Then nucleotide also means not only a single base like adenine guanine. Okay, sometimes the complete nucleotide excision may happen. Nucleotide means along with the sugar and a phosphate and with the nitrogen base. It will completely remove that nucleotide and it will add a new nucleotide. Then mismatch repair also. Okay, matching ATGC, right? So AT, so if it is not connected with the T, A with the G, okay, or G with the T, then that is a mismatching, right? So it will repair the mismatching also. Then double strand break repair means if there is any break in the phosphodiester bonds either in the single strand or in the double strand, then again it will repair. Okay, so after damaging, these repair may happen. Then transcriptional program activation also will happen. Sometimes if it is not uh, repaired, then what happens? It will send the information in the form of the mRNA as it is, right? Then mRNA will produce a different protein than the normal protein, right? Right, how the proteins are biosynthesized. How the proteins are biosynthesized based on information present on the mRNA. So how the mRNA will come? The mRNA will be coming from the DNA only. So when there is any DNA replication problem, so the coding sequence may get altered. So based on the altered coding sequence only, the mRNA is having the coding, right? So based on that mRNA coding only, the different type of protein will be synthesized. For example, you take the sickle cell anemia. Okay, a lot of diseases are there, thalassemia like that. So these are all like genetic defects only. So what is the genetic defects? Because that gene is not expressing the required protein. It is expressing a different protein. Got it? So, that is how, we get the disease. So transcriptional program activation may happen and it may lead to disease. Then cell cycle checkpoint activation also. Okay. So these are all the, this is just introduction. Now, coming to the, uh, actual content, DNA in the living cell is subjected to many chemical alterations, right? It will lead to uh, because when it is exposed to different chemical alterations, then automatically the DNA will get damaged. So genetic information encoded in the DNA has to remain uncorrupted because it has to pass from generation to generation. It should not be corrupted. Any chemical changes must be corrected. A failure to repair DNA produces a mutation. Okay. Then agents that damage the DNA. What are the agents? Just now we discussed. Highly reactive oxygen radicals. Okay, highly reactive oxygen radical, those are called the free radicals. Okay, I think, yeah, Tuesday you have a class. Okay, I think some DRP madam will explain this free radicals class. Then ionizing radiation, that are gamma rays and X rays, UV rays also, they cause the DNA damage. Then coming to the chemicals, aromatic hydrocarbons, plant and microbial products, aflatoxins. Okay, then when we use the chemicals in the chemotherapy, okay, especially to treat the cancer, all the cancer drugs are nucleic acid, works on the nucleic acids only, because cancer means it's a uncontrolled growth of a cell. How the uncontrolled growth of the cell will happen? The nucleic acid replication will happen. So most of the cancer drug will stop the replication. They act on the nucleotides only. Got it? So whenever a patient is taking a chemotherapy for any cancer, then even that will also damage the DNA. Okay. Then coming to the types of DNA damages. First, single base alteration. What do you mean by single base alteration? You know the strand, right? In the strand, the phosphodiester strand. I mean, that is a nucleotide strand. So in that nucleotide, if there is a change in one base, okay, then automatically that may affect the complete genetic code, right? So single base alteration. Sometimes the single base alteration may happen. How the single base alteration may happen? Maybe depurination. What do you mean by depurination? One purine will get deleted. D means removing, right? So what is the purination? What are the purines? Adenine, guanine, right? So either the adenine or guanine will be missed in that place. Okay, it got altered. Or deamination. What do you mean by deamination? Deamination also, it's a process, right? It's a process of removing the amino group from the cytosine, right? So when you remove the amino group from the cytosine, then it will be converted to the uracil. Got it? Even that is also problem, right? Because it will affect the genetic code, right? So in the place of cytosine, the uracil is coming. In the same way, and in the place of adenine, hypoxanthine is coming. So how these are coming? Simple. Deamination means if you remove the amino group from this base, then it will be converted to uracil. Then if you remove the amino group from the adenine, then it will be converted to hypoxanthine. Okay. It will result in abnormal base pairing, right? Then alkylation of base also. Later, insertion or deletion of nucleotide. Sometimes one complete nucleotide will get deleted, or sometimes a complete nucleotide will get added. Then base analog incorporation. Base analog incorporation is, for example, you know, right? AT, GC, right? So, I mean, this is the pairing, right? In the AC, I mean, AT, opposite pairing, right? But a molecule or any compound that is similar to the T, okay, it binds to the A, means that is an analog, right? It looks like a base. So that will get incorporated in the strand, but that is not the actual base. When it is the base, nitrogen base, then it will do the genetic coding, right? But if it is not base, if it is analog of base, then it won't be considered as a genetic information, right? Even that is also a problem, right? So single base alteration may happen. These are all the different examples, right? Then two base alterations may also happen. Not only single base, sometimes two alterations also happen. For example, you take the thymine. Okay, so because of this ultraviolet light, thymine dimer will form. Usually, adenine will be paired with the thymine, right? But here sometimes what happens, the thymine will pair with the thymine in a single strand. In the same strand, the thymine will get connected with the thymine. Means the base pair thymine will get connected, right? So that is called the dimer. I mean that particular word is called the dimer. What is the dimer here? Pyrimidine. That is a thymine uh, bond will form. That is called the dimer. Okay. So you have to remove the dimer. Okay. Otherwise, again it will lead to the DNA damage only, right? So we have to remove that one. Then bifunctional alkylating agents like cross linkage also will happen. Cross linkage means one is linked with the other one is linked with this one. Okay, the cross linkage also happen. Then there are chain breaks. Chain breaks means we have single strand and the double strand, right? So sometimes these ionizing radiation or radioactive integration of backbone element, oxidative free radicals also, chemicals also, they may break the DNA, okay, into pieces. The double stranded DNA, or sometimes it may also break a single stranded DNA also, right? Then there is one cross linkage. What is the cross linkage? Between bases in same or opposite strands, between DNA and protein molecules. Between, sometimes it may be interacted with the histone proteins also. Okay, that is called the cross linkage. So these are all the types of DNA damages. Clear? Now, coming to the yeah, so overall, okay, I'm just here showing the overall. See, this is a double-stranded DNA, right? Can you see here thymine dimer? In a single strand, this is the thymine, this is the thymine. So there's a link between these two thymine, right? So that's called the thymine or thymidine dimer. It's called the dimer. Okay. Then here there's a single strand break. Okay, DS double-stranded DNA break. Because can you see here the break is here and here. Two strands got broken. Right? That's the reason double-stranded DNA break here. So here a single strand break, single strand. then it's a mismatching. See, GATC, right? Here we have yellow color. Yellow means G. Okay. G should match with the C, but here it is showing A, right? So it's a mismatch. Okay. Yeah, this is a mismatching. Then covalent X linking. What's a covalent X linking? Usually the nitrogen base pairs should link it with the hydrogen bonds, not the covalent bonds. Is it covalent or hydrogen? I think you remember it's a hydrogen bonding, right? But here the link is with the covalent X linking. Okay, so that is also not good. You have to remove. Then there is an AP site. What do you mean by AP site? AP site. Here nitrogen base, right? Here also we have a nitrogen base. So here what is happening? Apurination. Apurination means the purine got deleted. The purine got deleted. So that is the reason this site is called the AP site, means apurination. Here the purine is not there. Then it has to add one more purine while repairing. Got it? So deamination. Deamination means here there is a cytosine. It got deaminated. Then it converted to the uracil. Okay. The uracil should not be here in the DNA. But here, because of this deamination of the cytosine, the uracil came. The uracil is not should not be here. It should be the RNA, right? But here the uracil is linked. So these are all the different types of DNA damages. Clear? Now coming to the repair. So already this we discussed. There is one direct damage reversal. See, when there is a damage, directly it will do the repair. There is no any problem. But sometimes it will do the excision. Means, see, when you see this part is damaged. Okay, this part is damaged. Then it will simply add this part here. Okay, it got repaired. Direct repair. Okay. So excision means here only this part got altered, right? So it is removing the complete that strand and adding a new strand, right? So nucleotide excision happening here. It's completely removing the nucleotide. Right? Then it is doing the repair. Okay, means most of the part it will excise, it will remove, it will cut, right? Then again in detail, coming to the direct damage reversal. Repairs damage directly without removing the nucleotides. The same thing, right? Without removing the nucleotides, wherever there is a deficiency of that nucleotide, it will just add, that's it. Okay. Repairs damage directly without removing the nucleotides. One example is photoreactivation. Usually this photoreactivation is absent in human beings. Okay. So what is happening? See, for example, when you're talking about the this thymine dimers. So if these organisms are exposed to sunlight, then automatically the sunlight will remove the bond between these two dimers, right? This is called the photoreactivation. What is that mechanism called? Photoreactivation. Okay. So what this photoreactivation is doing? Photoreactivation means there is an enzyme called the photolyase, okay, which will form by the sunlight only. I mean, it will get activated through sunlight. That's called the photolyase. Repairs thymine dimers using visible light only. Got it. It present in bacteria and plants, but not in the human beings. Means automatically the repairing will done. What type of repairing here? It will remove the thymine dimers. Okay. This mechanism is called the photoreactivation, and it's a natural phenomenon. Okay. Then coming to the direct damage reversal. The same one, right? This one, but I am showing with a diagrammatic representation here. So it's a native DNA. Here the dimers formed. Okay, this is the photoreactivating enzyme. So what is that enzyme called? Photolyase. So this photolyase is excising those dimers. Okay, dimer means like it is removing and then it is repairing. Right, release of enzyme to restore the native DNA. Okay, so this is called the photoreactivation process. Then excision of the, here we are talking about two methods. One is direct damage reversal. The second one is excision of the DNA damage, means damaged DNA it is removing, right? It is removing. So there are different methods. One is base excision repair, means it will remove only the base. Base means base pair. That is a nitrogen base, right? It will remove only base. Or sometimes it may remove the complete nucleotide excision also. Nucleotide means maybe 20 or 30 base pairs also, or 50, 60 base pairs also it will remove. Then mismatch repair. If there is any mismatch, then it will excise and it will add the new one. Then strand break repairs. Strand break repairs also may happen. What do you mean by strand break? If there is any break in the double strand or single strand, then it will repair. Right? In these reactions, a nucleated segment containing base damage, double helix distortion, or mispaired bases is replaced by the normal nucleated sequence in a new DNA polymerase who will repair the DNA, the damaged DNA. Polymerase only, right? DNA polymerase only. Okay. So all of these pathways have been characterized in both bacterial and eukaryotic organisms. Usually, all the mechanisms may not present in prokaryotes and all the mechanisms may not be present in the eukaryotes, and all these are not present in the plants and bacteria also, right? We are talking about the generalized things, means some mechanisms may present in prokaryotes, some are in plants. If you take the photo reactivation, that is happening, that's not happening in the human beings, right? So base excision repair, that may happen in human beings, but not in plants like that. Okay. So we are taking the, I mean, the generally, okay, it's not specific to one organism. So let's talk about the first one, base excision repair. How this base excision repair is happening? First, it will recognize, these are the steps. But first, it will recognize DNA glycosylase recognizes damaged base. Then base removed, AP site formed. What do you mean by AP site? Just now we discussed. That is here. What do you mean by AP site here? A purine got removed, right? A purine got removed. So that site is called apurinic site. A means absent, right? So this is a site where there is no purine. That's called the apurinic site. Clear? Clear. So that's called the apurinic site. So when there is any mismatch, it has to remove that one, right? So that is the reason here base removed. When the base is removed, then here it becomes an AP site. Okay, a puronic site. Then cuts the phosphodiester bond. Okay, there is an endonuclease, it will cut the phosphodiester bond because it has to cut the phosphodiester bond because we have to insert a new nitrogen base, right? So where the nitrogen base is linked to the phosphate only, right? So that's the reason they cut the phosphodiester bond, and then DNA polymerase action will happen, fills correct nucleotide, then seals the nick. So this is a base excision repair. Simple. I mean, you can imagine. Okay. So I'll explain. You can imagine there is a strand. This one mismatched base is there. You have to excise using this endonuclease enzyme. Okay. And then using the DNA polymerase, attach a new base, then ligase. That's it. Okay. Yeah. You see the example then. Here, C. Okay. C is there. Because of the heat energy or because of some mutation, here what has happened? Deamination happened. Deamination means C became the U. Okay, C became the U. In the DNA, U should not be there, right? So you have to excise the C. Then uracil DNA glycosylase. See here, which one? Glycosylase, DNA glycosylase. So here, uracil DNA glycosylase will removing that one, and it is converting this into AP site. AP site. Then nuclease is cutting that part. Then DNA polymerase is adding the nucleotides and then DNA ligase. So this is the base excision repair is happening. Okay. Where excision repair. Then coming to the second one. Second one is nucleotide. Complete nucleotide excision should happen. Right. This is also the same one. Okay. Just I will show the diagram here. Okay. Not only a single base pair here. We are talking about uh, maybe multiple nucleotides problem. Okay, means here it is a single one. Right? In the first one only base pair, only base, nitrogen base. But here bulky lesions, it's not one, bulky lesions. Okay, so damage recognition, again so this the same, endonuclease removes the damaged segment, then DNA polymerase fills the gap and ligation. Here multiple things, that's the reason nucleotide excision. Okay, the same thing. Again, the same example. Sometimes the dimers also. But here dimers with the photo reactivation, right? When you take the E. coli, what happens when there is a timer? Then in the E. coli, there are some proteins called UVRA, B, and C. So these will recognize the lesion. Lesion is the uncorrected part or damaged part, right? Always the lesions are nothing but the damaged parts. So here this is a damaged part or the dimer. What's the thymine dimers, right? So when there is any thymine dimer, then in E. coli, these three proteins, okay, identify the lesion and they will excise, and then DNA polymerase will synthesize a new strand, and these it get ligated, right? So, I mean, this happens, but when there is a problem in the nucleotide excision repair, means a person or a human being, if he is lacking this type of the mechanism in his body, then what happens? These are the features. Extreme UV sensitivity. Okay, so he will get a disease called the xeroderma pigmentosum. This is also an examination question. Xeroderma pigmentosum means on his face, uh, yeah, can you see? Yeah, this is called the xeroderma pigmentosum. Okay, so xeroderma pigmentosum is an autosomal recessive genetic disease. The clinical syndrome include marked sensitivity to sunlight with subsequent formation of multiple skin cancers and premature death. The risk of developing skin cancer is increased 1,000 to 2,000 times. The inherited defect seems to involve the repair of damaged DNA, particularly thymine dimers. Cells cultured from patients with xeroderma pigmentosum exhibit low activity for the nucleotide excision repair process. Got it? So, when the DNA collected from these patients, okay, and they worked on the excision process, but here the excision is not happening. Excision repair is not happening when you culture artificially after collecting the DNA from these people. Okay, so that indicates that if this mechanism is lacking, what is that mechanism? DNA excision repair. Okay, then there is a chance of getting this disease, xeroderma pigmentosum. Okay, yeah, but the thing is, see, for example, uh, okay, see, we are human beings, right? So you are taking a lot of, cancer causing agents, or you are exposed to high ultraviolet, or you are exposed to something, right? But they won't do any type of harm to you physically or phenotypically. Got it? Usually what happens, see, already the genetic, already the genes exhibited, right? So because of the gene sequence only, we bought into the world, right? So we have all the features just like our elders, right? But if genetic material altered in our body, so we are giving it to the next generation, right? So the next generation people may suffer. Got it? Because phenotypically these may not cause any problem for us, right? But when these are carried to the next generation, okay, then they may get affected. Okay, these xeroderma pigmentosum, all these the next generation may affect. Okay, not we. Okay, now coming to the mismatch repair. So what's the mismatch repair here? Mismatch repair corrects the errors made when DNA is copied. What, what is the DNA copied? When there is a replication, right? Replication. Okay, replication means creating a new DNA from the older DNA. At the time of creating a new DNA, when there is a mismatch, then that mismatch repair will be done by itself. For example, a C could be inserted opposite an I. Means in the place of A, C got inserted. Or the polymerase could slip or stutter and insert 2 to 5 extra unpaired bases. Means maybe in the place of C, A came, or in the place of A, C came, or sometimes, the flip, because of the flipping, 5 or 2 to 5 extra base pairs also they added. So specific proteins scan the newly synthesized DNA. I told you this is called a proofreading, right? It will check the newly synthesized DNA whether the replicated DNA is exactly the replica of the old one or not, right? Then using adenine methylation within a GT sequence as the point of reference, the template strand is methylated and the newly synthesized strand is not. Got it? Clear? So these things will happen. Means simple here, mismatch repair means, okay, let us see this. Single side strand cut means here it is not cutting the two strands, right? Because no need. Because this is the newly created strand, this is the old strand, right? So single strand cut by GATC endonuclease. Okay. Then defect removed by exonucleases. Okay, then it will synthesize. Simple here, mismatch repair means when the new strand is not matching with the old strand, right? Then it will do the repair. Okay, it is one example in the E. coli. But in the E. coli, these proteins are important. MUT S, okay, MUT L and MUT H. Okay, so these are the three enzymes. So here what happens if there is a problem in the mismatch repair? Okay, then this is an important thing to remember. Colorectal cancer and endometrial cancer. Okay, these are very important. This is called a Lynch syndrome. Lynch syndrome. Defective mismatch repair genes associated with colorectal cancer and endometrial cancer. Okay, then repairing strand breaks. Single strand break and double strand break. One is direct joining, it will do. Okay, one is the homologous recombination. Okay, it will combine the two chromosomes are there, right? So two chromosomes are homologous only, right? Then it will combine with the other chromosome. Got it? Either in the same chromosome or in the other chromosome. Okay. So here in the double strand break repair also, if it is not happening, then you will get this type of disease: Burkitt's lymphoma, B cell leukemia, Philadelphia chromosome. Okay. So yeah, so this is about the repair. Simple here, then there is a nick or when there's a break, so it has to do the repair. Okay. Then disease associated with the defective DNA repair system. Ataxia, telangiectasia. Okay, telangiectasia, Bloom syndrome. Okay, yeah, these are different diseases. Okay, let us see. The first disease, ataxia telangiectasia, AT, is an autosomal recessive complex multi-system disorder characterized by progressive neurologic impairment, cerebellar ataxia, variable immunodeficiency with susceptibility to sinopulmonary infections, impaired organ maturation, ocular and cutaneous telangiectasia, and a predisposition to malignancy. Okay, so these disorders may happen in this disease, AT. Then second one is Bloom syndrome. What is the Bloom syndrome here? Disproportionately small, the people. Okay, striking paucity of subcutaneous fat tissue throughout infancy and childhood. A redness of the cheeks and nose that characteristically makes its appearance in infancy after sun exposure. Chronic obstructive lung disease, diabetes mellitus, and malignancy of varied types are common complications of Bloom syndrome. Okay, then what is this? Cockayne syndrome. So this syndrome is also called the Weber-Cockayne syndrome or Neil-Dingwall syndrome. It's a rare autosomal recessive congenital disorder characterized by growth failure, impaired development of the nervous system, abnormal sensitivity to sunlight, and premature aging, hearing loss and eye abnormalities. All these things will happen. Okay, then trichothiodystrophy, brittle hair. Tricho means it's hair only, right? Brittle hair, rough skin, extreme photosensitivity are the characteristic features. Okay, the trichothiodystrophies are are named primarily for the hair sulfur deficiency, which is their most specific feature, which leads to brittleness of the hair. So there is a sulfur deficiency in the hair. So there is a deficit in DNA excision repair system along with other defects. Then Rothman-Thompson disease syndrome. So this is one. Then progeria. Okay, this is also one symptom. Then Werner syndrome. Okay, just go through this one. Then xeroderma pigmentosum. Okay, just if you remember the names, that's enough. No need to write elaborately, but just this one is important because I saw in the old test papers they asked about xeroderma pigmentosum. In a small question. Okay, so yeah, so this is about the DNA damage and repair. Okay, now let us see the mutation. What do you mean mutation? Any change in the DNA sequence of an organism is a mutation only. Okay, when the damaged DNA, if it is not repaired, then automatically the mutation will happen, right? So, what is this mutation? Okay, now we'll see the mutation. Any change in the DNA sequence of an organism is a mutation. Mutations are the source of altered versions of the genes that provide the raw material for evolution. Most mutations have no effect on the organism, right? Especially among the eukaryotes. Why? See, usually the mutations, see, if there is a small alteration in the gene, that may not affect us. Why? Because we have, a lot of genetic material, right? Human body contains, I think, 10 to the power of 13 cells our body contains, right? With 3 into 10 to the power of 9 base pairs, right? So a total of 10 to the power of 16 cell divisions will happen. So usually, for example, when you're talking about the mutations, mm, even 10,000 base pair changes also, they won't affect us. Why? Why? Because we have huge number of the base pairs, right? So even 10,000 also they won't affect us. Why? Because the complete DNA is not the coding area, right? We have a lot of DNA, right? But the complete DNA is not the coding one. Non-coding area also there, right? So the coding area is very less. So when you compare the DNA, the coding area is very less. What do you mean by coding area? Coding area means which synthesizes the mRNA. Then from mRNA, the proteins will come, right? So the complete DNA is not a coding area, right? Got it, right? So that is the reason when there is a mutation happens, okay, when there is any mutation happens, then it won't give you the abnormal protein because mutation may happen in the non-coding area. Got it? So that's why mostly it won't affect. Okay. So most mutations have no effect on the organism because a large portion of DNA is not in genes and does not affect the organism's phenotype. Of the mutations that do affect the phenotype, the most common effect of mutations is lethality, okay, because most genes are necessary for life. Only a small percent of mutations cause a visible but non-lethal change in the phenotype. Okay. Now, coming to the gene mutation. You know this one, right? Usually what happens, DNA sequence to mRNA will form, then from mRNA, polypeptides, means protein will form. Okay. See this example. So this is a DNA strand. Okay. GGT. So this is one genetic code in a normal gene. Okay. Then usually mRNA will be formed from the antisense of the DNA. I mean, the DNA will give the mRNA, right? In the process of first one is the replication. DNA to DNA is replication. This is called the transcription, right? So the mRNA will get formed from the DNA. mRNA formed from the DNA only. How it is formed? The mRNA chain contains the complementary. See, if it is G means it's having C. Okay. If it is G means C, T means A. So it's a complementary codons will be there, right? So we are taking, normal example I am taking here. Uh, any doubt? You want to ask something? No, right? Okay. Yeah, if you have any doubt, just type me in the meeting chat. Okay, I'll explain. Okay, so this larval gene, oh, sorry. Yeah, so this normal gene is transcribing, okay, this code to the mRNA. So based on the codes on the mRNA, it is providing you this amino acid like proline, okay, then glutamine, like cysteine, glycine, like this. Okay, we got these amino acids based on the genetic code. Okay, so here the antisense strand in the DNA. So this is called antisense strand DNA, which acts as a template for the mRNA. Then mRNA is giving this polypeptide. Okay, this is a normal mechanism, right? So normal gene. Then when there is a substitution, mutation means some change happened, right? When there is a substitution. Substitution means here. So this is a normal one. The same thing I copied here. So TC, in the place of T, A came. In the place of T, A came. Then what happens? Complementary to A is U only. Got it? So in the mRNA, instead of here A means here T. T means opposite should be A, right? But in the place of A, you came. You came. So actually one is proline. Okay. So glucose, glucose, cysteine. I mean glutamine, glutamine, cysteine, glycine. Okay. But here what is happening? Because of this genetic code, I mean triplet codon, here you can see the arginine came. Right. So substitutions will only affect a single codon. Here I mean it is affecting the complete code or it is affecting only single codon. Single codon only, right? See, the protein is same. Here the I mean the glycine is same, cysteine is same, everything is same. Here it is affecting only single codon, right? So because of this single base pair substitution, okay, it is affecting only single codon. Their effects may not be serious unless they affect an amino acid that is essential for the structure and function of a finished protein molecule. Got it? For example, see, it is one, it is only one. So it may not affect sometimes, right? See this example. Uh, yeah, here these are the codons. It's a normal one. In the place of T, in the place of T, okay, sorry, in the place of C, here T came. Okay, this is also giving you the, I mean, the glutamate only. Clear? It is also giving the glutamine. Got it? You know about the sense codons, nonsense codon. Do you have idea about that 64 codons? Yes, right. Sometimes, 3 or 4 triplet codons also will code a single amino acids, right? So here this triplet codon means GAA also giving you the glutamine, then GAG also it is giving the glutamine, right? So I mean this codon also giving the same, this codon also giving the same. Means though there is a change in the base pair, okay, the coding is same, means the same type of amino acids are you are getting, right? The same type of amino acids you are getting. Means sometimes though there is a mutation, the formation of the amino acid will be the same. Okay, but here substitution of only single codon. I mean, it is affecting a single codon. Here, there is no any change here. Means though there is a mutation, but there is any change. But here, when you see, this is a disaster. Disaster means here, in the place of T, sorry, here in the place of G, T came. Okay. When the T came here, okay, then opposite will be the A. Then what happens here? It won't, it's a nonsense. I mean, it's a nonsense codon. It won't give any amino acid, right? It won't code any amino acid. Then here it will get terminated. Terminated. Then instead of having the 5 chain, I mean 5 amino chain of a protein, so here it is terminating after the third amino acid only. So this is a disaster. So it will give a different type of the protein which may not required by our body. Okay, like this. So sometimes inversion may happen. Inversion means, see here, actually it should be see the second one, it should be CTC. But here inversion, in the place of T, C came, in the place of C, T came. So inversion, inversion. First place, second place, right? Then the second place come to the first place and first position coming to the second position. Okay, this is called the inversion. Even inversion also having, sometimes it may have a effect or sometimes may not. Sometimes the inversion also will give the same amino acid or sometimes it may give the a different amino acid. Right? That's called the inversion type. Then we have one more type called the frame shift. What do you mean by frame shift? Frame shift means here the complete frame is shifting. Got it? Here it is altering only one amino acid, right? Only one amino acid, remaining will be the same. But here the shifting is happening. What do you mean shifting? Shifting means here when you see GGT, okay, GGT, then CTC. But here after GGT, a G got added. G got added. So you know about the triplet codons, right? 3 will code one amino acid. So GGT will code proline. Then here GGT and later G got added, right? So actually, what is the second codon here? CTC. But when the G came here, it will become GCT. As there is no comma, right? It will become the GCT. So here CTC is coding the glutamine, but here GCT is coding the arginine, right? And what is the other one? CCT. Again, glycine, it's a different one, right? So here the complete frame got shifted, right? It's not like a single one. The complete frame got shifted by the addition of one nitrogen base. Okay. So here this is called the frame shift mutation. The complete frame is shifted. Okay. It altered the complete protein, right? Not only a single one, single amino acid. It completely altered the protein. So it gives a different protein completely. Okay. So this is the problem. Addition also, even the deletion also, not only addition, even the deletion also it will give the same thing. Deletion. Deletion means instead of CTC, right? So it is removing the T. Then again it will give a different amino, I mean, different protein, right? So classification of mutations are point mutations and frame shift mutations. Point mutation means it is defined as a change in a single nucleotide that may affect a simple or single amino acid. Right here, transition may happen, transversion may happen. Transition means a purine is replaced by another purine, or pyrimidine is replaced by another pyrimidine. That's called transition. Transversion means in the place of purine, pyrimidine came, or in the place of pyrimidine, purine came. That's called the transversion. Okay. Then coming to the frame shift mutation, addition or deletion. Okay, you see here transition. Okay, in transition means ATGC. ATGC means in the place of purine, another purine came. That's called a transition. Okay. So transversion. In the place of purine, pyrimidine came. In the place of pyrimidine, purine came. Frame shift means either addition or insertion. Okay. Then what is the effect of these mutations? Just now I told you. Silent mutation. Nothing will happen because though there is a change, it will give the same one. It will give the same one. See, when you see this one, what is happening here? Though there is a change, its mutation. Okay, as we have, see, 64 codons we have, right? 64 we have triplet codons. 64 are giving you only 20 amino acids. Means 2 or 3 also will give one amino acid only, right? So though there is a change in the substitution, okay, it is giving the same amino acid. So that is called the silent mutation. I mean, nothing is happening. That's called silent mutation. Okay, see this one. UCA, that is a serine. It's a normal one, right? But UCG, in the place of A, G came. So even UCG also, it will code the same serine only. So nothing will happen to the protein. It will be the same. So it's called the silent mutation. So what is the other one? Missense mutation. What is the missense mutation? See, UCA, right? This C is replaced by U. Then what happens? UAA, which is yielding a different one, different amino acid. That is a leucine. That is a missense only. Okay. Then here UAA, in the place of C, A came. Then it is a stop. Nonsense. Means it is stopping there, right? There's no any sense for this one. Silent mutation means substitution of the third base may not change the meaning of the codon due to degeneracy. Then, for example, the same thing I explained. Okay, missense. Okay. Then coming to the missense also, there are three things. See, we are talking about the missense, right? Missense means there is a sense. It is a completely silence. There's no problem because it's the same. Nonsense means it will stop. It will stop further adding up the amino acids to the protein chain, right? So it will stop. That's the nonsense. Missense means it will yield a different one, right? In the place of serine, it is giving the leucine. So that is a missense. But again, the missense acceptable because though it is giving a different amino acid, sometimes it may be acceptable, or sometimes it is partially acceptable, or sometimes it may not be acceptable. Okay, see for example, acceptable we'll see. Hemoglobin Hikari is an example of acceptable missense mutation because lysine at position 61 in the beta chain of is substituted by asparagine. Okay, in the hemoglobin structure, in the place 61 place of that hemoglobin structure protein, usually lysine should be there. Okay, in that B chain. But when the lysine is replaced by asparagine, okay, nothing will happen. Okay, the taking of the oxygen will be the same, everything will be the same. RB size will be the same, RB shape will be the same. Okay, oxygenation will be the same, everything will be the same. Okay, so it's a missense, nothing will happen. Okay, this mutant hemoglobin is capable of normal functioning. Okay, that is acceptable. But partially acceptable is, you know about this one, this is very important, right? Hemoglobin S. What is the hemoglobin S here? Substitution of glucose by valine at the sixth position, which may cause the, I think you know this one, sickle cell anemia, right? See, HbS functions normally at high oxygen tension, but get precipitated lower oxygen tension, right? That is the sickle cell. Will the people die with the sickle cell? No, they won't get proper oxygen. That's it. Okay, that is a partial acceptable. It's not like completely rejected, right? It's a partially acceptable. Okay, the shape of the red cell became sickle, but still they have the oxygen precipitation capacity, right? So that is the reason this is partially acceptable. And unacceptable means there is one more example. Hemoglobin M Boston is an example of unacceptable. Histidine at position 58 of a alpha chain is replaced by tyrosine. Okay, it is incapable of combining with oxygen. Then it won't get oxidized, or it won't having the capacity to take the oxygen or to hold the oxygen. Right? Then this is not acceptable. Then nonsense mutation means here it will stop. Got it? So nonsense mutation prematurely the formation of the protein synthesis will get stopped, right? So that is a problem. So there are some diseases called as cystic fibrosis, Hurler syndrome, okay, then Duchenne muscular dystrophy. So these are the problems. Then coming to the frame shift mutation. Just now we discussed frame shift means the complete frame is changing, means in the place of one amino acid, the other amino acid is getting created. See this example. The cat saw the dog, the cat ran. Okay. So here deletion of the, just we are deleting this H. 3 3 we have to read, right? The cat saw the dog, the cat ran. All the 3 3. So we deleted the H here. Then again after deleting the H, then you combine all the three. Then TEC, right? TEC. There is no meaning for the TEC. ATS, AWT, HED. Okay, we just remove this H and we I mean splitted them 3 3. So there's no meaning, right? Or you insert one more in the place of H, you add one more here. Then it will become THH, then ECA, TSA. There's no meaning for this one, right? So usually the frame shift mutations cause lot of effects, right? Because it will convert the, see, frame shift can convert a sense codon into a nonsense codon. This can cause premature termination of translation. Okay, there's no meaning, right? Conversely, a stop codon may be changed into a sense codon. An abnormally large protein may be formed as a result. So these are all the problems of the frame shift mutation. Okay. Yeah. So this is about the DNA damage and uh, repair, then clinical significance, and also